Antonio for this meeting. It looks like it's going to be a good one coming up. So first of all, as always, since we do get such sponsorship from the drug companies, please take a moment to visit the exhibitors. There will be a refreshment break between 3 and 3:30, and then a reception at 5:30 with drinks. You got a drink ticket and your name tag. And make sure you turn in your CMEs. There's practice management at the end of this talk tomorrow. For those who are wanting to stay, it should be very enlightening about Miraca. But we'll go ahead and get started. And so for the first talk, I'm proud to introduce Dr. Liu, who is the assistant professor, Department of Molecular and Human Genetics at Baylor College of Medicine. And he'll be talking today using the whole exome sequencing for technology for neurological testing. So please help me welcome Dr. Liu.
Thanks a lot for the nice introduction. And first, I'd like to thank the organizer for giving us the opportunity to present our work here. My name is Pengfei Liu. I'm faculty of Molecular and Human Genetics at Baylor College of Medicine. And I'm also one of the lab directors of Baylor Miraca Genetics Laboratories, specializing in exome sequencing. Today, I'm giving the talk on behalf of Dr. Christine Ying. And the topic of the talk today is the use of whole exome sequencing and other technologies in neurologic testing. For my talk today, I'll start with a specific neurological disease and talk about the development of molecular diagnostic technologies and their impact on the diagnosis in this disease. And then for the rest of the talk, and the majority of the talk, we will focus on clinical exome sequencing. Okay, this is a brief history of milestones in human genome analysis leading to clinical genomics. So the number of human chromosomes was only established in 1956 to be 46, and that is even after the elucidation of DNA double helix structure. And then following that, the first aneuploidy was described in the 1960s. And then with the development of the technology that they can define finer and finer molecular defects, in the 1980s, the first microdeletion syndrome was described, the Prader-Willi syndrome. And following that, in the 1990s, FISH was used widely, and that led to the discovery of many genomic disorders. So in the 21st century, which we call the post-genomic era, with the availability of the human genome reference sequence, chromosome microarray analysis, whole genome sequencing, and whole exome sequencing, these all started to get widely applied in clinics. So I'll start with this neurological disease, Charcot-Marie-Tooth, CMT, to talk about the development of diagnostic technologies and their implication in the diagnosis of this disease. So we all know that the majority of CMT patients, they are caused by a microduplication of human chromosome 17p12, encompassing the dosage-sensitive gene PMP22. So the concept of gene duplication leading to a disorder, this was the first one to be established in human. However, if you look back in the literature, in the 1920s, it was shown that trisomy can lead to a difference in phenotype in plants. And in 1930s, Dr. Kevin Bridges published this elegant paper in Science describing eye phenotype in Drosophila, where they see some flies have thinner eye than the other flies, and they call it a bar eye. And it was shown that this bar phenotype was due to duplication of a chromosomal segment using a Drosophila chromosome from a saliva chromosome from the Drosophila. Interestingly, they also note when they were screening for numerous bar flies that some flies had even thinner eye, which they call the ultra bar. And they were able to show that this ultra bar phenotype is because of a further increase in the dosage of that chromosome segment, which is a triplication that leads to an even severe thin eye phenotype. So this concept was clearly there in the 1990s when CMT duplication was first described. We were able to see CMT duplications, but whether triplication exists, whether they have a more severe phenotype than the duplication, they were all not known. And the first patient with CMT triplication was described almost 25 years later because development in technologies, for example, chromosomal microarray analysis, high-resolution CMA, MLPA, that were robust enough to discern the difference between a dosage increase to a duplication versus to triplication. So think about it, in the 1990s, we were screening millions of healthy individuals, and we were able to find a few patients with CMT duplication. And nowadays, with this massively applied clinical diagnostic technologies, we are able to screen thousands of patients with CMT duplication. And we were able to find that a few of them, they have a more severe phenotype. And we characterize them molecularly, they had the CMT triplication. So we know that CMT is a disease with great genetic heterogeneity, which means that there are many genes, different genes, in which mutation can cause a similar phenotype, CMT. So although the vast majority have the PMP22 duplication, other genes, mutation in other genes, for example, GJB1, MPZ, etc., they can all cause CMT. And nowadays, there are more than 50 known genes that are found to cause CMT. And many of these genes, they are discovered with the wide application of whole exome sequencing and panel testing, which lead to our major topic today, clinical whole exome sequencing. So for the rest of the talk, we will focus on clinical exome sequencing. And first, I'll introduce some basic procedure and concepts in clinical exome sequencing. And then we'll go into the details of our experience in the Baylor diagnostic lab, what we found by doing thousands of clinical exome sequencing. So we call this whole exome testing in the clinic a collaboration between physician and the lab. First, in the clinic, the physician and the genetic counselors will prepare the patients with informed consent and carefully describe their phenotypes. And this information will be transferred to the lab along with the patient sample, and we perform technical and analytical pipelines in the lab, and then interpret and validate the findings, and then draft clinical report. Finally, when the report is feedback to the clinic, that may be implemented clinically by the physicians. So the first step, pre-test counseling. The physicians will decide who in the family to test for whole exome sequencing. And in this process, we will tell the patient what to expect, what kind of results may come back. Some of them may be the diagnosis of the disease observed in the patient, and some others may be incidental findings, which means that there are diagnoses of a certain disease, but it's not the disease the patient came to the clinic for. And the patients will have the option to opt in or opt out, whether they want to know this finding or not. And I'll talk more about these incidental findings later. So it is critical that the disease phenotypes are described as detailed as possible and communicated effectively to the lab, because the whole process is a collaboration between the physician and the laboratory in finding the diagnosis. So to achieve that, we make a detailed questionnaire by organ system in which the physician can check and provide details which organ is affected or not. And in addition, more importantly, we request a detailed clinical note to be provided when the sample is submitted that explains the logic behind the exome testing and some differential diagnosis the physician may be considering. And there are numerous efforts in research toward adopting a structured language in making this communication between the clinician and the lab more and more effective, and this is still ongoing. So this is a workflow of whole exome sequencing. After the samples are received in the lab, we perform DNA extraction, and we perform a SNP array before putting the sample onto the sequencer. And that SNP array is for quality control step to make sure that the DNA is in good quality and to make sure that we have the correct identification of the patient. And then the sample goes through a library preparation, target enrichment, sequencing, and data analysis. Every step, we have QC of high clinical standard that sample has to fulfill. If not, they will go to the repeat until they fulfill this criteria. So this is an overview just to give you a flavor what kind of data analysis we are doing. After we get the raw data, we will perform alignment using different bioinformatics softwares, and then call SNPs, and then do various filter and quality control, and then finally the data will be fed to our LIMS system. So the data after this process we get is called the VCF file. That usually consists of many, many variants. On average, 200K variants. That's a lot of variants. And that has to go through first some quality filter and some filter whether these variants are observed in a general population or in any publication, and whether these variants affect protein structure by default, and what is the frequency in the population and in our internal lab. And finally, after all of these bioinformatics pipeline, we will come up with a list of about 400 to 700 variants that will subject to manual review. So this review process is performed by ABMG certified laboratory directors, and then the report is drafted by the directors and the counselors. And whenever we see a difficult case, we will have a meeting involving many medical directors and clinicians to discuss these difficult cases. And we also have monthly send-out meetings to discuss the most interesting educational or challenging cases. So exome sequencing comes in two flavors in our lab. First is Proband only, and second is trio sequencing. So in the Proband only sequencing, you do next generation sequencing only in the Proband. And for the family members, we will do Sanger confirmation for the interesting variants that will go, and the important variants that will go on to the final report. Whereas in the trio sequencing, we perform whole exome sequencing in both the Proband and the two parents. So in theory, we should have a similar diagnostic rate by either doing a Proband only or trio sequencing, because we are supposedly reporting variants in known disease genes. However, we are always interested in finding new disease genes and making new research discoveries, and that at the same time will result in increased diagnosis in the patient. In that sense, trio exome sequencing is a little better in discovering new disease, new diagnosis in novel disease genes, and thus may have a little higher diagnostic rate. So on our report, we report the variants in 5 classifications according to the ACMG guideline, from pathogenic to likely pathogenic, to variant of unknown clinical significance, likely benign, and benign. The first 3 will appear on the report, and the latter 2 will not appear on the report, but available upon request, because there are too many of them to be shown, and they are not very clinically significant for the patient. So they won't be on the initial report. And when we send the report to the physician, there is always a PDF form that is faxed to them, and there is also an electronic form of the report that have hyperlinks you can click, and that will bring you to the disease description, what is the population frequency, etc., which is more user-friendly. And as I just mentioned, we might find some incidental findings in the process of clinical exome sequencing. So some of the incidental findings may be medically actionable, meaning that we are very confident that these variants are disease-causing. In addition, there is direct clinically utility-based or established guidelines in the literature, or there is established treatment in the literature that if we report these life-threatening disease mutations to the patient, it may result in a benefit, even though the disease is not presenting at the moment. So some examples are Marfan syndrome, NF1, NF2, HMPCC, BRCA1-2 carriers. So as I mentioned, the patients have the choices to opt in or opt out. They can decide whether they want to know this or not before they start the sequencing in the pre-testing consolidation process. So next, we'll go into the detailed Baylor experience of whole exome sequencing in a clinical setting. Here are 2 of our milestone papers describing our experience in clinical whole exome sequencing. The first one was published in 2012 in New England Journal of Medicine, describing the very first 250 cases that gone through clinical exome sequencing. The second one is a later follow-up publication in 2014, published in JAMA, describing the subsequent 2,000 cases. This number is not correct, it's 2,000 cases. And right now, in our laboratory, we have over 8,000 clinical samples being sequenced by exome sequencing. And that only counts Proband, that doesn't count any parental sequencing or sibling sequencing in the trio setting. So the experience that we are going to talk about today will mostly come from this more thorough, this analysis including 2,000 samples. So the demographic breakdown of these 2,000 samples, the majority of them are pediatric patients below 18 years old. And the majority of them have neurodevelopmental disorders. For example, intellectual disability, seizures, developmental delay, and autisms. But there are also many other different kinds of genetic conditions, skeletal disorders, GI issues, etc., etc. Any kind of genetic disease that you can expect in the clinic. So this cohort represents almost no selection, no bias. And the patients come from a variety of referral sources from the academic or medical center, from within the United States, and from institutions, international, from other countries. So this summarizes the rough diagnostic rate that we found in this patient. So overall, when that paper was published, we have a diagnostic rate of about 25%. This number has changed when we do the analysis now, which I'll talk about later. And the take-home message of this slide is that if you break down the total number into different groups with different phenotypes, the group that has the highest diagnostic rate are those patients with a specific neurologic finding. For example, if there is a specific brain malformation, there is the highest chance to find a positive molecular diagnostic rate. And at that time, the rate is estimated to be almost 40%. And now we can say this number must be even higher. So this is a diagnostic rate comparison between Proband-only exome sequencing and trio exome sequencing. As I just mentioned, we had the option to only sequence the Proband or sequence all the 3 family members. So as expected, the trio exome sequencing have a little higher diagnostic rate, which is 33%, and the Proband only 28%. And the difference is because trio exome sequencing are better for discovering unknown disease genes. And for some of the patients who are critically ill, we have the option for them to do an expedited exome sequencing analysis, which we call critical exome sequencing. And if the patient goes through this route, we'll have a 2-week turnaround time. And in these patients, we found that the diagnostic rate is even higher than the other patients. And the diagnostic rate is about 50% in the critically ill patients. So these are the lessons we learned from doing the 2,000 exome sequencing. Among the positive cases, we found genetic mechanisms contributing to diseases of all types. Many of them are very rare genetic mechanisms. So we have de novo events, mosaicism, uniparental disomy, X-linked disease in females, consanguinity in autosomal recessive genes with a homozygous mutation, and autosomal recessive genes with a mutation, point mutation on one allele and the CNV on the other allele, and cases with 2 molecular diagnoses. I'll talk about this in detail in the following slides. So these are the summary of all the mutations in positive cases. This figure illustrates their mutation types. So we see that the vast majority of them, about 50%, are missense mutations. And then the remaining, almost the other 50%, are truncating mutations consisting frameshift, nonsense, and splice mutations. And there are also other types of mutations, in-frame, deletions, etc., of a smaller proportion. So if we break down the positive cases with their genetic inheritance model, about half of them are autosomal dominant. And among these autosomal dominant genes, 74% of the mutations arose de novo. And the inherited portion is much smaller. So this is one of the examples that we learn from doing this, which is different from what we were expecting according to the textbook. So for the autosomal dominant conditions, what we learn from the textbook is that there is usually a family history expected. However, what our experience told us is for the majority of them, there is no family history. 74% of them came de novo. And some other patients can even be mosaic in the parents, or in the de novo sense. There's no family history. So for autosomal recessive conditions, what the textbook told us is that they usually come in compound heterozygous in an outbred population. Our experience in this 2,000 sample is that we have both compound heterozygous point mutations and homozygous mutations in patients with shared ancestry in their parents. But surprisingly, we also found some examples where one of the allele has a point mutation and the other allele has a CMV, usually a deletion in it. Another patients are homozygous for the mutation, but it's not because that the parents have a shared ancestry. Instead, this homozygosity arose by uniparental disomy. So for X-linked diseases, what the textbook told us is that we expect many more males than females. But our observation from these 2,000 cases is that they're about equal. They're about 50% in males and 50% in females. And this is the concept of dual molecular diagnosis. So what this means is that sometimes in the patients that we found a positive diagnosis, there is not only one mutation. There are two mutations or even 3 or 4 mutations in some extremely rare cases. And these mutations will be in 2 or more genes that sometimes have overlapping phenotypes, and some other times they have completely different phenotypes. So the diagnosis is a mixture of these 2 diseases. And we observed consistently among different groups that among all the patients with a positive diagnosis, there's around 5% of them with a second or more molecular diagnosis. So here are some examples of patients with 2 diagnoses. The concept of blended phenotypes and 2 diagnoses is really critical because what we have been told in doing molecular diagnosis and clinical diagnosis in genetic disorders is that we should be looking for unifying diagnosis. However, in these patients, there is no unifying diagnosis. We have to be able to split these diagnoses in order to make sense what is happening in this patient. For example, this first patient, if whole exome sequencing was not performed, let's assume that this was done 10 years ago. The patient came into the clinic with ataxia telangiectasia kind of phenotype and with some mild spastic paraplegia phenotype. And the physician decided to do a Sanger sequencing on the ATM gene and found the mutation in the ATM gene. Then one may start to think that this is an example of clinical variability in which they found a novel mutation in ATM that not only caused ataxia telangiectasia, but also caused some other phenotypes like spastic paraplegia. But that is not correct until you perform exome sequencing that in an unbiased way survey the entire genome of all the genes, and then you found that there is a second mutation in spastic paraplegia, and you know that this is a patient with a dual diagnosis. Oh, I forgot to mention that this is very critical concept to us because sometimes these patients present a mixture of two phenotypes, which is why they remain undiagnosed, because the phenotype doesn't look like either of the disease. And these are usually very challenging cases, and the diagnosis is very difficult until you apply exome sequencing on them. So here is our experience in incidental findings. As I just mentioned, we occasionally find incidental findings, and if they are medically actionable, we have the possibility to report them if the patient opts in to know these results. So about 5% of all the patients we tested have a positive incidental finding. And some of these are in genes that are recommended by the ACMG to be the medically actionable genes. And some other genes are not in the ACMG recommendation list, for example, G6PD, etc. But we still think there is clinical utility if we tell the patient that they have this disease. So we might report those as well. Here is an example of successful implication of exome sequencing result on medical management of a patient. So we have a 38-month-old female with static encephalopathy, hypertonia, and seizures. Exome sequencing identified a de novo mutation in the SLC2A1 gene, which encodes glucose transporter 1. And mutation in this gene, patients with mutation in this gene, ketogenic diet is highly effective in controlling seizures for these patients. So that was done after the patients know they have this mutation, and there was dramatic improvement of the movement for this patient. So even though the majority of our cases are pediatric, we also have about 10% of them who are over 18 years old, which we consider adult patients. So we think that this group is interesting, and we wonder why these patients remain undiagnosed until they are adult. And we looked at this group of patients, and we found that some of them is just a very rare type of disease. For example, this disease, which is only been seen in the literature for 20 cases. Some other times is that the patient has a classic genetic syndrome, but the phenotype is milder, it's not classic. So this is a kind of phenotypic expansion. For example, we have a patient with Soto syndrome, but the patient had no overgrowth. so they remain undiagnosed until they're adult. Also, we have examples in the adult patients, similar to in the pediatric patients, that we found specific treatments or patients with blended phenotypes. So one eternal topic that a molecular diagnostician can always think of is we want to improve the diagnostic rate. And our experience told us sometimes you don't even need to repeat the experiment. You can just do some reanalysis, reinterpretation over time, come back to the case and do a reanalysis, and you get a very significant increase in the diagnostic rate. So why is this? This is partly due to the nature of exome sequencing. When we perform a clinical exome sequencing, we generate lots of data, and we only analyze a small fraction of them, only in the exome and only in the genes that we know. And our knowledge of the human genome is rapidly evolving. There are constantly new disease genes being published and new experiments being done for novel variants. Additionally, the patients may have a clinical phenotype that keeps evolving. And as time goes on, they may have more clinical phenotype that if they provide that update to us, we'll have a better idea what kind of genetic disease they may have, leading to an increased diagnosis. So here are the consequence of reanalysis performed on these two cohorts. The first cohort corresponds to the 250 cases in our New England Journal paper, the very first 250. And the second cohort consists of the following later 2,000 cases from our JAMA paper. At the time these two papers were published, the respective diagnostic rate was a similar 25%. If we perform reanalysis now, the diagnostic rate jumped to 36% in the first group and 30% in the second group. If we zoom in to the increment of this diagnosis and see why we are making new diagnoses, we'll notice that the majority of the increment are because of new disease genes, which mean that at the time the case was initially reported, the actual mutation is in a gene that was not known to cause disease yet. But later that gene is published and we can make the diagnosis. So here we plotted each case as a dot here, and the x-axis indicate the original date of release date of their exome report, and the y-axis indicate the time of the publication for their actual disease gene. So for example, this patient here has their exome report was initially released in 2012. However, the actual disease gene was published in 2016. So at the first time, we couldn't make a diagnosis. But after the publication came out in 2016, we can issue an addendum report and finally make a diagnosis for this patient. So for all the cases above this diagonal line, their mutations are residing in the gene that were not even published yet at the time their initial exome report were released. So if we assume that we are performing our reanalysis in 2014, we would make this more new diagnosis. If we do it in 2015, we can make more diagnoses. If we do it today, there are even more. Assume that we do this in the future, I expect that we still have more and more diagnoses coming because there is still constantly new paper being published for new disease genes. Skip this. Okay, so if we come back to this image and focus on the rest of the graph, we'll see that besides the contribution of new disease genes, we have contribution from many other reasons that we are making new diagnoses. For example, the green one, CMV analysis, that is making the second highest contribution. And we note an interesting trend that in the earlier group, CMV analysis is only making a small contribution to the new diagnosis. But as time goes by, it seems that the contribution is growing. So why is this? Our interpretation is that this probably means that exome sequencing is becoming more and more popular. And sometimes, as I just mentioned, the patient is so ill that they don't have time to wait for CMA. So they submit exome sequencing right away. That's why we are catching these diagnoses which could have been caught in a CMA, but as our technology analysis algorithm improves, we can now detect some of the CMVs in exome, and we can make these more diagnoses. So finally, I'd like to talk about the new disease gene discovery activity in our lab. Because we are in an academic institution, and all our laboratory directors are faculties of Baylor College of Medicine. So we always have strong interest in identifying new disease genes. And we have several mechanisms in which we put through our cases that had an initial negative diagnosis from clinical exome testing, and then put them through a research mechanism. And these very frequently result in discovery of new disease genes. These are some of the examples. And here are some of the publications that came from our laboratory and from this research mechanism. Some of these genes turn out to be one of the most frequently mutated genes in any exome lab. For example, the Pura, the Tango2, we have 17 cases with Pura mutation in our lab, 14 cases with homozygous, with biallelic Tango2 mutations in our lab. So one of the research mechanisms is CMG, Center of Mendelian Genomics. This is a multi-center, collaborative, huge effort with the two major centers in Baylor and Hopkins, and also with centers in Yale and University of Washington, with the emphasis to discover new disease genes. So we'll put many of our undiagnosed cases through this mechanism to see if they can identify new disease genes and thereby increasing our diagnostic rate. The second research mechanism is UDN, Undiagnosed Disease Network. This is also a nationwide, huge collaborative effort, and there are multiple clinical sites, 7 clinical sites and 2 sequencing cores. And Baylor Genome Sequencing Center is selected as one of the sequencing core. And we perform the analysis in the diagnostic lab. So the objective of UDN, similar to the CMG, is to improve the level of clinical diagnosis and care, especially in challenging cases, in undiagnosed cases. And at the same time, facilitate research and with the interest of identifying new disease genes. And also to facilitate integrated collaborative effort between research and the clinic in a more translational way. So in summary, we have seen strong and growing interest in whole exome testing in different clinical scenarios. We have tested pediatric neurological phenotypes, adults, and many other patients in our clinical exome lab. We have found diagnosis in many rare conditions and common conditions, and we have a positive diagnostic rate of around 30% in a highly unbiased, unselected clinical population. And as I've shown, there is a slightly higher diagnostic rate for trials compared to Proband sequencing. And we have shown early evidence of clinical utility and cost effectiveness in applying clinical exome sequencing. And finally, I've shown that the non-diagnostic cases can go through several research mechanisms, potentially finding new diagnoses and describing new disease genes. So this is a group of Baylor and BMGL exome team. We are very proud of our team because of these extremely talented and diligent people working to make this team and contribute to the diagnosis for patient care. Thank you for your attention. Any? Well, right now is, I believe it's 7,000, but there has been effort, a lot of effort to bring that.
It's your face I see, my inner light. What could it be?
Thank you very much. Okay, so moving right along, our second speaker today is Dr. Gelfand, who comes from us all the way from University of California in San Francisco. He did his medical school at Harvard, so went from one coast to the other and is now visiting the middle. So hopefully he has specialized in MS, neurosarcoidosis, and autoimmune encephalitis, and I think the first talk is on autoimmune encephalitis, so please help me welcome Dr. Gelfand.
Thank you so much for the invitation to be here. I'm excited to be in Texas and to discuss two topics that are very close to my heart and that I'm excited to share some updates with you about. And I tried to make these talks as